photosynthesis is perhaps the best engineered sequence of events in nature. This sequence of complex chemical events in the biological world is responsible for the maintenance of all life forms. It involves mainly two parts, light reactions and dark reactions. Here we will focus on the light reactions and comparing the electron transport in photosynthesis to a relay race. This video will bring to you the most precise information using animations which you will never forget. Because of its complexity, there are a lot of misconceptions surrounding light reactions. This video tries to make those concepts clearer, and clearer. Hello everyone, it's me Shanty and you're watching Biology Nowadays. As we all know, photosynthesis is of two kinds, oxygenic and anoxygenic. But what's the difference between these two kinds of photosynthesis? Oxygenic photosynthesis involves the production of oxygen and anoxygenic photosynthesis does not involve the production of oxygen. Oxygenic photosynthesis is seen in cyanobacteria, green plants and algae, while the anoxygenic kind of photosynthesis is seen in primitive photosynthetic organisms such as green sulfur bacteria and purple bacteria. Even though there are two kinds of photosynthesis, when we just say photosynthesis, it normally refers to oxygenic photosynthesis because of its great importance in sustaining life on Earth. As I told in the beginning of this video, the process involved in photosynthesis can be divided into two series of reactions, light-dependent or light reactions and light-independent or dark reactions. The light-dependent reactions, as the name indicates, depend on light directly. These reactions use light energy to produce ATP and NADPH. ATP or adenosine triphosphate is a cell's primary source of chemical energy. It is the universal energy currency of life. The other product, NADPH or reduced nicotinamide adenine dinucleotide phosphate, is a cell's primary source of reducing power. In the light-independent or dark reactions, ATP and NADPH produced in the light reactions are used to reduce carbon dioxide to glucose. During this process, the carbon which was present in the atmospheric carbon dioxide is now fixed in solid organic compounds, for example, glucose produced in plants. So this process is called as carbon fixation. The dark reactions are also called Calvin cycle, which we will discuss in a following video. A point to be noted here is that even though dark reactions are called so, they normally occur in the light because they require the products of light reactions which are ATP and NADPH. The word dark just indicates that light is not directly involved in these reactions, but actually they are indirectly dependent on light. Light reactions take place in the granule and stromal thylakoid membranes of the chloroplast, while the dark reactions take place in the stroma, which is the fluid matrix inside the chloroplast. If you are not so familiar with the structure and components of chloroplast, I highly recommend you to go back to part 1 of the photosynthesis lecture series before you proceed. Light reactions are also called primary photochemical reactions or Hill reaction, as this was discovered by Robert Hill in 1939. As you already know, it takes place in the granule and stromal thylakoid membranes. In the part 1 of the photosynthesis lecture, we had learned about the photosystems and how light energy is transferred to the special pair chlorophyll A molecule in the reaction center of each photosystem. In this video, we will see the further steps involved in the light reactions. That is, what happens after our special pair chlorophyll A molecule receives light energy. Light energy triggers the flow of electrons through a series of molecules from one end to the other. And it's quite interesting to learn. It's like passing a baton from one runner to the next runner in a relay race. Through the hands of many runners in the ground, the baton reaches from the starting point to the finishing point. Here in photosynthesis, the baton is the electron. Now let's get familiar with the players in the ground of photosynthesis. Unlike in the case of real relay race, here in our photosynthetic relay race, some players don't need to run, but they just have to pass the baton to the next player standing next to them in the lane like this. Here the playground is the thylakoid membrane. Let's have a closer look at the thylakoid lipid bilayer membrane. Most of the players belong to two different types of photosystems, photosystem 2 or PS2 and photosystem 1 or PS1. PS2 is mainly localized in the closely packed regions of granule thylakoids. PS1 is found in the stromal thylakoid membranes and in the grana margins that are exposed to stroma. Here in this picture, I have shown them closer just to make the explanations easier. PS2 also consists of an oxygen evolving complex which helps the water to split into H+ ions or protons, oxygen and electrons. An important player in PS2 is the special pair chlorophyll A molecule, which is referred to as P680. Here P stands for pigment and 680 means 680 nanometer. This is the wavelength of light that this special pair chlorophyll A molecule absorbs most strongly. The next member of PS2 is the pheophytin molecule. Pheophytin is a chlorophyll A molecule without the central magnesium ion. Attached to PS2, another important player is there, and that is plastoquinone. Between PS2 and PS1, an enzyme called cytochrome B6F complex is present. The next player is plastocyanin. Plastoquinone and plastocyanin are mobile electron carriers. It means that they can accept the electron, move some distance and hand over the electron to another acceptor, just like a runner in a relay race hand over the baton. Plastoquinone is a fat-soluble protein and moves in the thylakoid membrane between PS2 and cytochrome complex, while plastocyanin is a water-soluble protein and moves in the lumen between cytochrome complex and PS1. In PS1, a special pair of chlorophyll A molecule is present and it is called as P700. Again, P stands for pigment and 700 means 700 nanometer. This is the wavelength of light that this special pair chlorophyll A molecule in PS1 absorbs most strongly. Remember, in PS2, instead of P700, it was P680. The special pair chlorophyll A molecules in the reaction center of PS2 and PS1 differ in their light absorption maxima because of the differences in their protein environment. In addition to P700, PS1 consists of the next player named A0, which is a modified chlorophyll molecule. Outside PS1, the next player comes ferredoxin, which is a small iron sulfur water-soluble protein found in the stroma. And after ferredoxin, a large enzyme called ferredoxin NADP plus reductase or FNR is present. FNR is found mainly in stroma and sometimes either loosely or tightly bound to the thylakoid membrane. It is also reported that FNR is found bound to the inner membrane of the chloroplast envelope. There is also another enzyme called ATP synthase. ATP synthase is seen in the stromal thylakoids or stroma lamellae and also in the edges of granule thylakoids. This enzyme is not involved in transferring electrons. Instead, it transfers the H+ ions or protons from lumen to stroma and makes use of the proton motive force to produce ATP molecules. During the electron transfer, the electron generated from water splitting by oxygen evolving complex is passed to P680, which is then passed on to all the players one by one and reaches the NADP+ and result in the production of NADPH. The direction of electron transfer in photosynthesis is from PS2 to PS1. Now you may be wondering why the first photosystem where photosynthesis is initiated is called PS2 and the later participant as PS1. There is a reason for that. In fact, the second one was discovered first, and so it got the name PS1. Okay, let's come back to our discussion of electron flow. We are going to look in detail the electron transfer mechanism from one molecule to another. To start with, you may just suppose that I isolated the chlorophyll A molecules from a piece of green leaf. Chlorophylls are fat-soluble pigments, so I used an organic solvent, for example ethanol, to extract the chlorophyll A molecules, and now I have with me a conical flask containing chlorophyll A molecules dissolved in ethanol. In the chlorophyll A molecule, the electrons are arranged in their molecular orbitals in such a way that the molecule has the minimum possible energy, and at this stage it is said to be in the ground state. When a photon hits the molecule, the energy of the photon is transferred to one of the electrons so that the electron jumps to a higher energy orbital. At this stage, the chlorophyll A molecule is said to be excited. Excited states are unstable and will return to the ground state by the emission of light, which is called fluorescence. Remember that this is observed in isolated chlorophyll A molecules dissolved in organic solvents such as ethanol. Now here comes a question. When leaves are exposed to sunlight, the chlorophyll A molecules in the leaves will also get excited. Then why don't the leaves show fluorescence? Interestingly, fluorescence is not the path chosen by our chlorophyll A molecules in the leaves to come back to their ground state. It's a quite different process, and that's the key to photosynthesis. And what's that? Here is the special pair chlorophyll A molecule P680 in PS2. By absorbing light, it will get excited. Our chlorophyll A molecule will now give off energy by emitting an electron. Unlike in the case of isolated chlorophyll A molecules, there is a primary electron acceptor near to the special pair chlorophyll A molecule when it is present inside a photosystem. This primary acceptor will accept the electron, and because of this, the leaves don't show fluorescence. The primary electron acceptor close to the special pair chlorophyll A molecule in the PS2 is pheophytin. Pheophytin accepts the excited electron coming from the special pair chlorophyll A molecule. Now, the chlorophyll A molecule gets a positive charge. But let me ask you a question. Why chlorophyll A molecule should be excited by sunlight before it can transfer the electron to pheophytin? In other words, why pheophytin cannot accept an electron from a chlorophyll A molecule which is in the ground state? In order to understand this, let us learn a little bit of electrochemistry. Don't worry, it's simple. Here P680, the special pair chlorophyll A molecule of PS2, is in the ground state. Now, suppose that P680 loses an electron and becomes P680+. The process of losing an electron is called oxidation. The tendency of P680 to become P680+ is called its oxidation potential. Let's draw an axis here, indicating the standard oxidation potentials in volts. The oxidation potential increases upward along the axis. In some popular textbooks, you may find standard reduction potentials on this axis. In fact, oxidation potentials and reduction potentials have same values but with opposite signs. I have chosen oxidation potential here for making the explanation a little easier. So, the standard oxidation potential of our P680 plus redox couple is negative 1.1 volt. Now let's consider the situation when light falls on P680. It becomes excited, which is indicated as P680 star. If P680 star loses an electron, it becomes P680 plus. Now we are considering a new redox couple P680 star, P680 plus. The oxidation potential of this couple is 0.7 volt. Remember, a species with higher oxidation potential will have a greater tendency to lose electron. In other words, they are better reducing agents. So, who is the better reducing agent in this case? P680 or P680 star? Obviously, P680 star is the better reducing agent because it has a higher oxidation potential. Now let us consider pheophytin, which is the primary electron acceptor in PS2. By accepting an electron from P680 star, pheophytin becomes pheophytin minus. The process of gaining an electron is called reduction. This pheophytin minus and pheophytin is another redox couple. The oxidation potential of this one is 0.6 volt. A species up in the scale can reduce the species down to it. So who can reduce pheophytin to pheophytin minus? P680 or P680 star? And yes, the answer is P680 star because its oxidation potential is higher than that of pheophytin redox couple. P680 cannot reduce pheophytin because its oxidation potential is lower than that of the pheophytin couple. So without light, P680 is unable to give an electron to pheophytin to start the photosynthesis process. Amazingly, Sun does this great job by exciting P680 to P680 star and thereby increasing its oxidation potential and making it capable of giving an electron to pheophytin. This is a wonderful design of nature. Now, there is a problem. The electron has reached pheophytin, but P680 has become P680 plus. It has to come back to P680, the ground state form. For this, it needs an electron, but who will donate the electron? The electron will come from water, which is abundant in nature. A water molecule will split into two H plus ions, an oxygen atom and two electrons. Actually, splitting of water is not an easy process. If we want to split water in a lab, then we have to use strong electric current or high temperatures, nearly 2000 degrees Celsius. But in our plant cell, the oxygen evolving complex in PS2 will do this job by utilizing the energy of photons. This light-dependent splitting of water is called photolysis. The oxygen atom formed by splitting of one water molecule is now within the oxygen evolving complex. The oxygen evolving complex is situated towards the lumen side. So, the lumen of the thylakoid is the site of water oxidation. The electrons from water will go to P680 plus and P680 plus will get reduced to P680, the original ground state form. It means that water reduces P680 plus, or in other words, P680 plus oxidizes water. As I said earlier, the oxidation of water is very unlikely to happen in the normal course. But P680 plus does this. So P680 plus is considered as the strongest biological oxidant known. Now let's see what happens to the electron received by pheophytin. Pheophytin donates that electron to plastoquinone or PQ. At this stage, PQ will accept an H plus ion from stroma. In fact, PQ is a 2-electron acceptor unlike other electron acceptors in the chain. So from where it will get the second electron? The answer is that all the electron transfer mechanisms that we discussed just now from water to pheophytin will happen once again. That is, the light excites P680 to P680 star and its electron is accepted by pheophytin to become pheophytin minus. At the same time, P680 star becomes P680 plus, but it immediately accepts the second electron from water and comes back to P680, the ground state. Pheophytin donates that electron to plastoquinone or PQ, and then PQ will receive its second electron. Along with the second electron, it accepts a second proton, obviously from stroma. In short, PQ receives 2 electrons and 2 H plus ions and now it becomes hydroquinone, PQH2. In many popular textbooks and diagrams, you might have seen that electrons move in pairs. In fact, no. P680, pheophytin and all other electron carriers except PQ are 1 electron carriers. In these carriers, the electrons are not moving in pairs but 2 times, 1 by 1, just like we saw now. PQ is a mobile carrier and it moves to cytochrome as hydroquinone. Hydroquinone passes the electrons one at a time to plastocyanine or PC through cytochrome B6F complex. When one electron passes from hydroquinone to PC, one H plus ion is liberated to the thylakoid lumen. Remember that PQ had accepted 2 H plus ions from stroma while accepting 2 electrons from pheophytin. Now when it loses these electrons, the H plus ions are also liberated, not back to the stroma but into the thylakoid lumen. In addition to these H plus ions, 2 more H plus ions are also pumped from stroma to lumen. Why these 2 additional H plus ions are pumped? Because the electron transfer from PQH2 to PC takes place through cytochrome in a cyclic process called Q cycle. And absorption of 2 H plus ions from stroma is required for completing the Q cycle, and then the H plus ions will be liberated into lumen. I will explain the Q cycle in another video. So here, in effect, 4 H plus ions are pumped from stroma to the thylakoid lumen, while PQ transfers the electrons to PC through cytochrome complex. Together with the 2 H plus ions from the photolysis of water, now there are a total of 6 H plus ions in the lumen. This will create a concentration gradient of H plus ions between stroma and lumen across the thylakoid membrane. It means that stroma has a shortage of H plus ions while lumen has excess. The H plus ion concentration gradient is then used by the enzyme ATP synthase to produce ATP from ADP and inorganic phosphate. But how? The H plus ion buildup in the thylakoid lumen generate a chemiosmotic potential across the thylakoid membrane. Due to this, the H plus ions pass back to the stroma, driving the ATP synthase. In fact, ATP synthase is a machine. Yes, a molecular machine, described as world's tiniest rotary motor working by the flow of H plus ions or protons through it. In other words, by a proton motive force. Here, I have prepared a model of ATP synthase. It consists of mainly a head, CF1, which contains the catalytic site of the enzyme where ATP is produced, and a base, CF0, which mediates the proton movement. CF0 and CF1 are connected by a rotary shaft. The flow of protons causes the rotation of the CF0 subunits, which will result in the turning of the rotor shaft. Covering a portion of this rotating part, there is another subunit, which I have not shown in this small model, and it is connected to CF1 by another structure. If I cut the top part of the head of the ATP synthase, you can see the rotor shaft with a small pointer-like structure. The rotation of the base results in the rotation of the rotor shaft, which in turn brings about conformational or structural changes in the head CF1 subunits. These conformational changes are responsible for the production and release of ATP molecules. A full rotation of the rotor shaft results in the production of 3 ATP molecules. And for a full rotation, an average of 12 H plus ions should pass through the base CF0 subunits. In other words, 4 H plus ions should pass through ATP synthase from lumen to stroma for the production of a single ATP molecule. The process of formation of ATP from ADP and inorganic phosphate in photosynthesis is called as photosynthetic phosphorylation or photophosphorylation. See here, a series of electron transport process initiated by sunlight resulted in the migration of protons from stroma to lumen, creating a chemiosmotic potential across the thylakoid membrane, which pushed the protons through the ATP synthase from lumen back to stroma and resulted in the production of ATP. What does it mean? The plant cell has harvested the light energy and it has been stored as the chemical energy in ATP. This ATP can be converted back to ADP and inorganic phosphate so that the stored energy can be released. Almost all life forms on this planet Earth utilize this energy exchange method, and so ATP is called the universal energy currency of life. So it's not merely boost, the secret of my energy, but it is ATP too. Now let me share an interesting fact about ATP. The ATP content in an average human body is about 50 grams. but the daily energy requirement of the body is up to 75 kilograms of ATP. If our body has to carry this much ATP for its daily function, we all will be like a big sack of ATP. But fortunately, the 50 grams of ATP present in our body is recycled again and again to meet the daily energy requirement. One ATP molecule is recycled up to 1500 times in a day in a human body. Now let's come back to the discussion on electron transport. Our electron is now with PC. PC is located in the thylakoid lumen. It is water soluble and so it moves between cytochrome complex and PS1 through the thylakoid lumen. In PS1, we have the special paired chlorophyll A molecule, that is P700. PS1 receives a photon and it causes P700 to jump to its excited state P700 star. Now P700 star donates one electron to the modified chlorophyll molecule A0, a primary electron acceptor which is situated inside the reaction center of PS1. At this time, P700 star becomes P700 plus and it comes back to the P700 ground state by receiving an electron from the plastocyanin. PC leaves PS1. Now what happens to the electron accepted by A0? It is transferred to the ferredoxin, a small water-soluble iron-sulfur protein which is in the stromal side of the thylakoid membrane. This electron is then transferred through the enzyme ferredoxin NADP plus reductase or FNR and finally reaches NADP plus. NADP plus can undergo 2 electron reductions and so it needs one more electron. But where's the second electron? You may remember that one of the electrons from PQ is still in the cytochrome complex. It is now passed to plastocyanin, then to P700 molecule in PS1 after the excitation by a photon. Now A0 has accepted an electron and this electron will be transferred to ferredoxin and to NADP plus through FNR. So NADP plus receives a total of 2 electrons and by combining with an H plus ion from stroma, it produces NADPH. Now I hope you clearly understood how 2 electrons are being passed from a single water molecule through a series of electron carriers to NADP plus to produce an NADPH molecule along with ATP. But are these transformations energetically feasible? Let us have a short look at the graph showing oxidation potentials of different chemical species involved. We already discussed this graph until pheophytin. By donating an electron to pheophytin, our P680 star has become P680 plus. It will be reduced to P680 ground state by accepting an electron from water. Are you wondering why I did not show the arrow of electron transfer to P680 plus written here? Note that by accepting an electron from water, P680 plus becomes P680 ground state and not P680 star, which is in the excited state. So P680, P680 plus is the correct redox couple involved here. You may have to recall one point that I told in the beginning of explaining this graph, that a species above can reduce a species down to it as determined by their oxidation potentials. Here is the position of PQH2, PQ couple in the scale of standard oxidation potentials. So it is obvious that pheophytin minus can reduce PQ to PQH2 and PQH2 can reduce PC to PC minus, catalyzed by cytochrome B6F complex. During this step, the concentration gradient of H plus ions created across the thylakoid membrane is used up by ATP synthase to produce ATP molecules. And PC minus will reduce P700 plus to P700. And stop. Now we will have a tendency to say that P700 will reduce the next electron acceptor A0 to A0 minus. But is it possible from this graph? No. Because P700 plus redox couple is below the A0 redox couple. So, electrochemistry, doesn't allow the reduction of A0 by P700. But sunlight comes to help the electron to continue its journey. On receiving a photon, P700 is excited to P700 star, and the position of P700 star, P700 plus redox couple is now above A0. So the job is done. P700 star reduces A0 to A0 minus. Remember that during this time, P700 plus will accept an electron from Pc minus. Now A0 minus reduces Fd to Fd minus, and then Fd minus gives its electron to NADP plus through FNR to finally form NADPH. Now let us look at the shape of this graph covering the events from one photoexcitation to the other. They lie under a Z-shaped path. So the pathway of this electron flow is called the set scheme of photosynthesis. Now we know what happened when one water molecule is split. 4 photons were harvested, that is 2 photons by each photosystem. 1 NADPH molecule is formed. 6 H plus ions came to lumen, 2 from the water molecules and 4 from stroma. So how many ATP molecules will be formed? Remember, 1 ATP requires 4 H plus ions to pass through the ATP synthase. So 6 H plus ions can generate, let's say, 1.5 ATP molecules. But see, one oxygen atom which is produced from water is still in the OEC, or oxygen evolving complex. It is not liberated yet. Now another photon hits the P680 molecule in PS2 and it gets excited and gives off an electron to pheophytin. At the same time, a second water molecule is split and the whole electron and proton transfer mechanisms we discussed so far repeat once again. The oxygen atom produced from the second water molecule combines with the already existing oxygen atom in OEC and gets liberated as an oxygen molecule, which will diffuse through the thylakoid membrane. The oxygen molecule escapes through stomata and finally reaches the atmosphere. Please note that for the ease of explanation, sometimes I have let one electron to stay in some molecular species for a relatively long time until the other electron has reached far ahead. In fact, the electrons are flowing from one molecular species to another in a continuous fashion. Since a large number of water molecules are split in every second, a large number of electrons become available to run through the electron transport system. When an electron enters one end of the electron transport chain, another electron leaves from the other end. It is like a train. When the engine pulls the first coach by 1 meter, the force reaches the last coach almost simultaneously, which will also be pulled by 1 meter. You just saw that a complete reaction involves the splitting of 2 water molecules and the harvest of 8 photons. The reaction produces 2 NADPH and 3 ATP molecules. Let us write the overall light reaction as 2H2O plus 3 ADP plus 3 inorganic phosphate plus 2 NADP plus and 8 photons give 3 ATP plus 2 NADPH plus O2. Thus, in the light reaction, there is a continuous flow of electrons from water to PS2 to PS1 to NADP plus. The electron ejected from PS2 doesn't come back to PS2. The flow of electrons is unidirectional. So this is called non-cyclic photophosphorylation. As you can see here, the ATP to NADPH ratio is 3:2. But sometimes the ATP produced in this way may not be sufficient for the plant's requirement. When there is a high demand for ATP, the photosynthesis system adopts another mechanism of electron transfer, and that is cyclic photophosphorylation. In the part 3 of this lecture, we will see cyclic photophosphorylation in detail. Let's conclude this video about the amazing process of photosynthesis with a quote from the famous Nobel laureate Albert Szent-Györgyi. What drives life is a little current kept up by sunshine. Thank you for being with me and stay tuned.